incubate.
Hi, we're in the back room where the incubation is going to happen. The dishes are here. They've been prepared and sealed. 3 of them are done. I'm just going to show you how to do one of them to show you the technique. And then the incubator is over here. This is basically it's a tank of water. It's just a cooler. There's a pump and an aquarium heater in there. This is set at a temperature of 44.5 degrees. As I pull this out and read it, 44.5 degrees, what do you know? That's a temperature, it's a little warmer than body temperature, but it's a temperature that fecal coliform grows really well at. And we put them in water because it's just a very stable environment, and they're going to stay at the same temperature. They're going to grow. They're not going to be in the water, but the water surrounding the dishes will keep these at a very steady temperature. So what you want to do with these dishes, it's a little tricky. You want to seal the edges of these with electrical or black tape. So I'm just going to chop a piece off. It's a little tense with the gloves on. It's kind of a pain because you got latex gloves and plastic tape, but you can do it. And again, if you have a classmate back here helping you, 2 heads are better than 1, or 2 sets of hands are better than 1. Make sure as you wrap it around, you're just sealing the edges of the dish, because we don't want water actually getting in there. These are going to be underwater, but they're not going to come in contact with the water, hopefully. And if you press the tape on the edge of the dish, you should be good to go. So you're going to take all 4 of them now. You may have a bag smaller than this one, but whatever you have is fine. You want to put them upside down, so the purple part of the dish is showing. The bacteria grows better that way. You just put them in a couple of stacks like this. And when you seal the bag up, I'm just pressing the bag, and I'm getting as much air out of here as possible, because we want the bag, we don't want it to float, we want it to sink. So I'm going to seal it up. You're going to label your class on here. There might be 3 or 4 sets of these in there, and even though your dishes are labeled, in order to prevent any chaos over here, just make sure you label your bag. Flip it over. You can just fold the plastic. And now to make sure, I'm going to put this all in here, but to make sure it stays under the water, I'm going to put a weight on top of all the dishes. This is just a scuba diving weight, or like a weight belt, so it's just going to stay on top of these for a day while they incubate. When you open the lid here, there's a mercury thermometer in here. Make sure you take this out and put it on the side table. Handle it carefully. We don't need it broken. You can pop the lid off, and you're going to see and feel a whole bunch of warm water in there. So I'm going to put my bacteria down here. These will definitely float if I don't weigh them down. I'm going to take my weight, put it on top. And when I come back in one day, 24 hours from now, you may have to come in, if you're on an A day, you might have, you or one of your classmates have to come in on the B day. You can't let them sit in here for more than a day, or it's going to mess everything up. So make sure you talk to your classmates and your teacher and arrange who's going to come in here and check these a day from now. And so the next thing we'll see, we'll pull these out of here one day from now, and we'll show you how to analyze them and what kind of results to look for.
Okay, it's been 24 hours since we put the fecal coliform bacteria into the incubator. And so this is one of the things that your group's going to have to do. At least one member of your group's going to have to come back 24 hours later, and hopefully we'll do this test. We usually try to do this test so it's not on a Friday, because coming in on a Saturday would be a bit of a hassle to come in. But let's say that your group did this on Wednesday. You'd have to come in on Thursday even though you don't have biology, so you'd have to try and figure out a time that's as close to 24 hours after the time you tested to get your Petri dishes. And again, you're going to have to wear your goggles and gloves because we're actually working with bacteria here, and so we want to be very careful. And I've taken the 4 Petri dishes out of the incubator, and you can see that we've got fairly decent results here. Here's our control, and we would expect no bacteria in our control, and we don't have any. There's nothing growing there. Interestingly enough, at 0.1%, we also don't have any bacteria there. Well, you think about that. That river water's really been diluted down. There's only, a fraction of actually 0.1% of river water in that. And so there were no colonies that showed up there. When we get to 1%, however, you're going to start to see some blue dots. Those are the actual bacteria that got trapped on the filter paper and then with the food medium grew and developed to be large enough to be visible with the naked eye. So you can see those pretty easily, and they just show up as blue dots. If they aren't blue dots, then you know that it's a different type of bacteria, because the way that the food is treated, it causes the bacteria to grow as a blue dot. So you can see that one bacteria over 24 hours became 2, 2 became 4, 4 became 8, and so on, so that after 24 hours we can see enough of them. Now, so far that data is good. Then we get to 10%, and oh my gosh, we got a lot more bacteria there. You can see them scattered around. And that's what exactly you want to see when you get your bacteria. That's the whole point of swirling when you did the millipore apparatus, the filtering apparatus, so that the water would be distributing the bacteria across the filter evenly. And that's exactly what we saw. Otherwise, they could all be localized into one spot. And when you get that, you know you've got some problems. So here we have our results. And what you'll do is you'll write down in your lab notebook, hey, how many did you have? And the beautiful thing about our results is it progressively increases, and that's what you want to see. You want to see that there are more bacteria in the 10% than in the 1%, and more bacteria in the 1% than in the 0.1%. And hopefully your control has no bacteria in it. Otherwise, you know you had some sort of failure in your sterilization process. So now that we've examined our Petri dishes and collected them, give them back to your teacher, and your teacher will destroy the bacteria so that there's not a chance of letting anything out that could be harmful to us. And now you're ready to calculate what the overall number of bacteria means in terms of water quality. Okay, now we have our results from our fecal coliform bacteria. And what I've done is I've put it in a form that you can see a little bit easier. These circles represent our Petri dishes. And once again, just to go over our results, you can see for the control we had no colonies present, which is great because there were, in fact, not supposed to be any colonies present. We used deionized water. There shouldn't have been any bacteria in that. And our results indicate that there weren't. Another good bit of news is that at the 1/10 of a milliliter of river water, okay, put into our filtering apparatus, showed no colonies present either. That's a good sign. That means that there's not a lot of bacteria in the water. Then we step it up to 1% of water, and we had 5 colonies present. And you can see these little blue dots that I've drawn here. They represent the fecal coliform colonies. Everywhere there's a little spot is where one bacteria got trapped on the filter, and then after a 24-hour incubation period in the fecal coliform food, these bacteria multiplied enough so that they appear, there's actually millions of bacteria there, they appear as a shiny blue dot on the Petri dish. So this is an actual colony initiated from one bacterium being trapped on the filter paper. Finally, we get to the 10%, and we see that there were 34 colonies present. After I counted them on the grid, we saw 34 colonies present at 10% river water diluted with 90% deionized water. So then what does that mean in terms of the overall water quality for us? Well, the next thing I'd like to show you is that based on our results, we did things right. some of the problems that you can run into when you do this filtering of your river water is that, for example, you didn't get an even distribution of bacterium around the filter paper. So that means that the growth around the sealing edge was an unclean filter holder or a poor seal. Somehow it didn't work out right for you. So one of the things that you want to look for is to see that you've got an even distribution. Here we've got a dry spot, which means you didn't seat the filter properly, or the shaking and swirling did not take place properly. Here, this would happen if we use, for example, 100% river water. The sample size was just too large. The colonies actually fuse together, grow together, and you can't count the individual colonies of bacterium, so you end up with a problem there. And here again is uneven distribution from not swirling the sample correctly or not adding distilled water to the filter sample. Down here is what it should look like, and this in fact was what ours did. It was between 20 and 60. We had 34 bacterium at our 34 colonies at 10%, and here we saw that if it's done right, you usually end up between 20 and 60 fecal coliform colonies, and they're evenly dispersed throughout the filter paper. Okay? So we were pretty excited about our results. They made sense, again, because the actual results went and gradually increased. None, a few, and then even more. So it makes a lot of sense that as the concentration of river water increased, the number of bacteria that we trapped on the filter increased as well. Now what does that mean? 34 colonies present. Well, the next thing that you do is in your water quality analysis handbook, there is a something called a Q value or a quality value. The Q value looks at quality for that particular water test that you do. So in this case, we've got the fecal coliform colonies, 100 milliliters over 100 milliliters of water. All right? So I had to do a little mathematical tinkering here. If there was 34 colonies present at 10%, we have to kind of extrapolate and say, hey, what if it was 100% river water? And in this case, it would be just times 10. Okay? For example, if you got 5 colonies at 1%, it would be 500 at 100% river water. So here we got 34 colonies at 10%, 340 colonies at 100%. So what do? Well, down here, it indicates on the x-axis what the number of colonies per 100 milliliters of river water would be. And in this case, it was between the 200 and 500, 340. I kind of estimated or kind of guesstimated where that 340 would be. Okay? I slightly kind of cut the difference right there, and I think I did a fairly decent job. And then here is your Q value. So your Q value is just basically a score out of 100. Really low amounts of bacteria would be high water quality, and you'd have a higher score. Really large amounts of bacteria would not be a good indicator of water health, and so you would have a low Q value score. Well, what you will do is, based on the number of bacteria you found, the bacteria colonies per 100 milliliters of water, that best worked out for you, you will follow that up until you intersect this pre-drawn line on the graph. In this case, we were at 340. We came up, and it intersected right there, and we came across, and we saw that our Q value score was right around 35. A Q value of 35 out of 100 isn't necessarily a great score. I mean, you wouldn't run home to tell your mom and dad, hey, I got a 34 on my final or 35 on my final. That would not be a good score. Likewise, it is here not a good score. If we only had picked up 5 or 10 bacterium per 100 milliliters of water, hey, that's a pretty good score. Because remember, the bacterium reflect that there's some sort of pollution getting into the water, an excess of waste from warm-blooded animals, either be it birds or humans or dogs or cattle. Something is applying fecal coliform bacteria to the water, which indicates a chance for a more harmful pathogen, say cholera or typhoid fever, being present in the water, which could be a serious problem in terms of overall water quality. So Once again, you just find the number, you follow it up, see where it intersects, come across, and that gives you your Q value for the water quality for fecal coliform. And then you're ready to start writing your report and getting ready for your oral presentation that you'll give to the rest of the class, indicating what you found out about fecal coliform and the Chicago River. Good luck.